Okay, hello everyone, and welcome to this Integrated DNA Technologies webinar on improving next-generation sequencing target capture. My name is Hans Packer, and I will be serving as moderator for today's presentation. The presentation today will be given by Ibrahim Jivanjee, and Ibrahim has a bachelor's degree in molecular biology from the University of California, San Diego, and a master's of business administration from the University of Iowa. His background is in product-focused research and development in proteomics and diagnostics and therapeutics. Ibrahim currently works as a product manager at IDT, and he's assisting in the development of next-generation sequencing solutions. Today, Ibrahim's presentation should last about 20 minutes, and following that presentation, Ibrahim will be joined by Dr. Adam Clore, who is a scientific applications specialist at IDT. And then Ibrahim and Adam will answer any questions that you have about the presentation. As attendees, you've been muted, but we encourage you to ask questions at any time during the presentation, and you can do that by typing them into the questions box, which you'll see located at the right-hand side of your screen. And if you click on the little plus sign, you can make that bigger so you can type into it and kind of even click on the blue bar and drag that out if you want to make it a little bigger. At the end of the presentation, then I will direct your questions to Ibrahim and Adam. In case you need to leave the webinar early or you want to revisit this webinar later, we're recording the presentation, and that will be available on our website at www.idtdna.com. And you'll find that under our support tab under the technical and educational materials. We also will be posting the webinar on our YouTube channel, which is at www.youtube.com/IDTDNABio. And there's a host of other webinars there on things like qPCR and synthetic biology, so you might want to check those out. So at this point, I'm going to turn the presentation over to Ibrahim so he can get started. Ibrahim? Maybe? Ibrahim, are you there? Ibrahim, we can't hear you. We seem to be having some technical difficulty here. Hang on a second.
Hello, test.
Ah, there we go. Gotcha.
All right. Sorry about that. Thank you, Hans. Sorry about the technical difficulties. As Hans mentioned, my name is Ibrahim Jivanjee. I am the product manager for next-gen sequencing applications for IDT. Today, I will be talking to you all about improving NGS target capture with xGen lockdown probes. And with that, I'd like to get started. So just to give you a brief overview, I'm going to start with a high-level overview of the next-gen sequencing workflow, and then highlight where IDT hopes to add value. And then I will talk more specifically about target enrichment and its benefits, along with some challenges with doing enrichment. And finally, I will introduce xGen lockdown probes and the benefits and challenges that we hope to address. So getting started, the general next-gen sequencing workflow begins with genomic DNA, which is then sheared to 100 to 300 base pair fragments, and which are then purified. Adapter and barcodes are attached onto these fragments, and then they are enriched as an optional step, followed by template preparation, which is an attachment to beads or solid support, such as the flow cell for Illumina or to beads for 454 ion torrent in solid. Those library is then sequenced and then sent on for analysis. Here at IDT, we want to focus on two areas: adapter and barcode attachment and enrichment. Specifically for this talk, I will be talking about enrichment for target capture of next-gen sequencing. So what is target enrichment? This graphic highlights two applications for next-gen sequencing, which is whole genome sequencing or targeted resequencing. The gray shaded region shows whole genome sequencing, where you have fragmented library with adapters attached and then sent for sequencing. Generally speaking, you can do 1 to 10 samples of whole genome sequencing in one sequencing run. The target size for that application is generally 3 gigabases and is usually useful for discovery or building a reference genome. Targeted resequencing, on the other hand, can be done either with hybrid capture or amplicon generation before sent for sequencing. You can increase your sample throughput to hundreds to thousands of samples, and the target analysis size could vary from 5 kb to 60 megabases. The applications used for targeted resequencing are rare variant discovery and variant detection, and other applications exist as well, but those are the primary ones. So more specifically, what is the value and benefit of enrichment? This example provides various scenarios based on a HiSeq 2000 run, and we assume a cost of roughly $20,000 for one sequencing run. So the first area that we want to focus on is throughput and cost. As you can see with human genome sequencing, you can do around 10 human samples at roughly $2,000 per sample, just for the sequencing itself. If you try to increase that coverage to 500x, which is useful for rare variant detection, you would not be able to do that with human genome sequencing given current technology. However, with human exome sequencing or a custom 500 kb capture, you can increase your sample throughput for a 30x coverage up to 200 to 40,000 samples. And if you are doing rare variant detection or variant discovery, you can do 12 to 2,000 samples. So your throughput is greatly increased as you enrich for your sample for a more relevant target sequence. Furthermore, your cost is decreased. You can go from $2,000 per sample to less than $1. The next benefit of doing enrichment has to do with the applications that you can potentially do. Particularly for diagnostics, custom captures or human exome captures are more suitable for doing that. And for rare variant detection, it is absolutely essential that you enrich so that you can increase your coverage and be able to detect those variations more effectively. So why is increased coverage important for rare variant detection? This graphic shows the required coverage given an expected mutation rate. Expected mutation rate's on the x-axis. It declines as you go to the right, and coverage is on the y-axis. The expected mutation rate is a function of the sample homogeneity and mutation frequency. So as an example, if you're doing cancer sequencing, when you take a tumor biopsy, you rarely have 100% pure tumor sample. You generally have we can have up to a 50% tumor cells with the subclones within those cells, which cause you to have to detect for a rare allele within that cell population, which then calls for increased coverage required. So some of the challenges with enrichment methods include design. For example, there are no standards for best practice for design, and there's very little data for best practices, which results in a need for trial and error. Optimization and expansion is another issue in which current designs that you purchase today tend to be static. So modifying them after you purchase them tends to be difficult. Balancing sensitivity, specificity, and uniformity is also a challenge. Sensitivity refers to percent of on-targets captured. Most researchers require 100% of their targets captured, and it's required if you want to reliably detect or discover rare variants. Specificity is also an important metric, and it refers to percent on target. It's important when you have limited reads. However, if you're doing sequencing on a HiSeq platform or GA2X, one of those high throughput machines, you generally have excess sequencing capacity. So percent on target isn't as important for some customers, but when you have a benchtop sequencing platform, it tends to be more important in those situations. Coverage uniformity is also an important metric in which you measure how tight sequencing reads vary within the average of the mean coverage. So low uniformity can create sensitivity issues, and enrichments with poor uniformity would result in regions that are over-sequenced with regions that are under-sequenced. So to compensate for those regions of low coverage, you have to over-sequence. Workflow is another challenge. Hybrid capture workflow can be complicated and time-consuming. Amplicon sequencing workflows can be limited in what they can detect, and they also suffer from biases, which I will cover in this next step. So hybrid capture biases often suffer from, are often TM related, such as %GC content of your targets. Amplification methods suffer from multiple amplification biases in which you can have over or underrepresentation of mutations based on random statistical occurrences. And then the final challenge has to do with scalability. For most researchers or clinical customers, it's difficult to achieve economies of scale due to the high upfront costs required and the high volume of samples required to achieve economies of scale. So now we have IDT xGen lockdown probe technology. Our lockdown probes are 100% QC'd with ESI mass spectrometry. We can pull up to 2,000 of these probes per tube, and our standard and Excel scales can be delivered in plates. Our product is shipped within 7 to 10 business days of order. These probes are all individually synthesized and all contain a 5' biotin modification. There are 3 scales available: the mini, the standard, and XL. The mini scale yields 2 picomole per probe with a magnitude increase as you increase in material per probe as you increase your scale. The minimum order for the mini scale is 200 probes, so you can potentially develop a panel using the mini probe scale for a 5 kb region. And then the amount of captures that you receive increase as you increase the number of probes you purchase. So for the mini scale, that's equivalent to 0.15 captures per probe. And for the standard, it's 1.5 and 15 for the XL. So as you increase the scale, you can increase the number of captures by either purchasing more probes or increasing the scale of the probe that is synthesized. Some of the challenges that we address with xGen lockdown probes are optimization and expansion. You can potentially mix and match custom pools according to your research needs, and you can supplement an existing pool to improve enrichment performance. We also address uniformity with a 10% improvement in uniformity versus a capture using array-derived RNA baits. Sensitivity is also addressed by ensuring that virtually 100% of target bases are captured. And biases are reduced with our QC process. So sequence-related biases are reduced with individually synthesized and QC probes. One of the main differences of our technology versus other capture technologies is that we individually synthesize and QC our probes, thereby ensuring that each individual sequence that is synthesized is present in your pool before it is delivered to the customer. DNA probes are also more forgiving to TM differences. So as you go beyond 60 bases, you generally have a TM profile that evens out. And finally, scalability. With our lockdown probe technology, you can achieve economies of scale when evaluating a small capture region. So what are some of the results of our lockdown probes? First thing that we can do is augment the performance of an existing capture. In this example, you can see a coverage. This is a coverage chart, and this is a 1.1 megabase capture using RNA or array-derived RNA baits. As you can see over here, there's a region which is severely underrepresented in this capture experiment. Now compare this with the performance of this capture experiment below. This is the same 1.1 megabase capture with the only difference of supplementing xGen lockdown probes to enhance the coverage of that region that was poorly represented before. So one of the most popular applications of lockdown probes is to improve the capture of existing capture panels that you can purchase today from such technologies as NimbleGen or SureSelect. Another important benefit of xGen lockdown probes is improved uniformity. Here we have a coverage map with normalized coverage along the x-axis, 1 representing the average coverage, and percent of total coverage represented on the y-axis. The first thing that I'd like to show you is that the xGen lockdown probes follow a more standard curve versus RNA capture, which tends to be skewed on the lower end. xGen lockdown probes have 53.4% of sequences covered within plus or minus 0.2 of the mean. The RNA capture set had 44% of sequences covered within plus or minus 0.2 of the mean. Furthermore, the RNA capture resulted in more sequences with reduced coverage when compared to our xGen lockdown probes. This can result in wasted sequence coverage if you have a hard minimum coverage requirement for your panel. Another benefit of xGen lockdown probes is the ability to create very small custom panels. So in this graphic, we have a 133 kb capture and a 29 kb capture sequenced using a HiSeq 2000 49 by 49 base pair read sequencing run. The 133 kb capture had 99.9% of sequences covered at greater than 500x. This is enough coverage to detect rare variants with an allele frequency of 5% with high reliability. The 29 kb capture has a 99.9% of sequences covered at greater than 750x. So it's amazing that you can achieve such high level of coverage with a capture panel that is so small. Another important benefit of using xGen lockdown probes is the modularity and the flexibility of the product itself. Unlike array-derived capture panels, xGen lockdown probes are individually synthesized and QC'd, which means that you can create small panels individually from each other. So for example, if you started with a 200 kb capture panel and you added in a 70 kb capture panel that was synthesized independently and a 30 kb capture panel, you can have a new panel that is 430 kilobases. This is important for applications in which you start with a conservative design and optimize as you need based on your sequencing needs and your research goals. The second example is something I showed you earlier, in which you have an exome capture panel that is supplemented with xGen lockdown probes to improve the performance of an existing panel. Before, you would have to synthesize an entirely new array of probes just to compensate for the inadequacies of that design. Now you can just supplement that capture panel with our probes to improve performance. Over the years, IDT has developed a reputation for quality, reliability, and speed. This is no different with xGen lockdown probes. With our 100% QC, we provide you with mass spec traces for each and every single probe that we synthesize. This mass spec information is shown in the an example of mass spec information is shown in the left graphic over here. And then we also provide OD measurements for each and every probe that we synthesize that is available as an Excel spreadsheet that you can download once you purchase your product. And that's shown over here in the screenshot. Another important attribute of our product is that the probes can vary in length between 60 and 120 nucleotides. This accommodates very custom, unique designs that for enriching for very difficult regions of interest. For example, some customers have targets that have repeat regions interspersed throughout it. So you can design lockdown probes to tile against your target regions, which are shown in green over here, using variable length probes, 120 nucleotides at 2x coverage, while completely avoiding your repeat regions. Avoiding repeats is important because repeat regions can result in low specificity of capture. Because 50% of the genome is repetitive, tiling against a repeat can potentially capture sequences that are non-specific to your regions of interest. And finally, one very important benefit of lockdown probes is the scalability. In this example, we show a potential pipeline for customers doing clinical applications in which they develop a panel using our mini scale with a limited number of samples. validate that panel over time with a standard scale, and optimize it to improve performance, and commercialize it. So whether you have 30 samples or 30,000 samples, Lockdown Probes has a scale that can meet your needs. So in summary, next-gen sequencing enrichment technology selects specific genetic regions of interest for deeper and more affordable analysis. This allows for greater sample throughput at a lower cost. Furthermore, deeper sequencing allows for the ability to identify rare alleles. Some of the challenges with enrichment technologies today include low coverage and uniformity, and xGen lockdown probes improves uniformity and depth of coverage, as well as allows for scalability and higher levels of quality controls. With that, I would like to conclude this presentation. Additional resources are available at www.idtdna.com/pages/xgen, which is our product catalog page for our lockdown probes. We also have a decoded article highlighting highlighting Foundation Medicine, who use our product and did an interview with us to show how well our lockdown probes worked for them. This is decoded 2.3, and the article is called Genomic Target Selection Using Individually Synthesized Capture Probes. Finally, if you have questions or requests outside of this presentation, you can email me at ijivanjee@idtdna.com or email xgen@idtdna.com. With that, I would like to take any questions that you may have.
Okay, I want to thank Ibrahim for that informative presentation, and now I'm going to welcome Dr. Adam Clore to the discussion. So welcome, Adam. And we will take any questions that you have. If you haven't already done so, you can type your questions into the questions box, which is located at the right-hand side of your screen in the GoToWebinar software. And all you have to do is click the plus sign. You'll get a space to type in your question. And we have a couple of questions already, so we'll just get started with that. So the first question, Ibrahim, is does IDT have any of their own exome capture panels?
We do not have our own exome capture panel at this point. We encourage customers to work with the panels that are available commercially today. Those panels will definitely have holes in them or coverage gaps, and we encourage customers to fill in those gaps with our lockdown probes.
So do the customers have to design the panels, or do they get those panels from somebody else? Here's a different question. Do we provide any help designing the panels?
Oh, definitely. So if you have regions of interest that were not covered appropriately with the design that you already had, if you send those sequences to us, we can tile probes for you and synthesize them for you. So the information that we would need are the regions of interest that were not covered appropriately, or for your custom panel.
Okay. Let's see here. So next question is, this question isn't. All right, I'm going to ask a different question first. So how does using the xGen lockdown probes affect quantification of gene expression if you're doing a exome capture?
I think that's an important question to ask, and it's something that is very important to consider when you're doing enrichment. Anytime you do an enrichment, the process of enriching is going to introduce some bias into the sample that you have. There's always the chance that you will not be enriching for all sequences equally. And so that can affect the way that you're calculating the quantification of your material. There are some people that will not use any enrichment techniques for quantification, and there's some people that are comfortable using some enrichment techniques. It really comes down to the level of specificity that you need in the specific organism and specific DNA that you're working with.
Okay. Adam, I'm going to actually give this question to you as well. Can this technology be used to specifically identify RNA viruses that are in a clinical sample?
You could. We've worked with several customers who have been interested in enriching for specific pathogens within a host organism. So as long as you know the sequence of the organism that you would like to enrich for, or in this case a virus that you would like to enrich for, you can use an enrichment technique to increase the number or increase the relative amount of the sequence in there. Now with RNA viruses, it's a little more challenging because of the high rate of mutation that occurs within these replicons, and so you want to make sure that you're using a sequence that is targeting the majority of the viruses that are present in the organism that you want to enrich for.
Okay. There's two questions here that are kind of related about working with, like what genomes do we support with the xGen lockdown probes? And one of them goes a little bit into detail. They're saying that they're working with a serial, and they want to know if it can be applied to other organisms. What should they do to work with xGen lockdown probes in this organism?
So just so I'm correct, they're working with a serial grain. Is that?
I'm guessing yes. They're not entirely clear on that. I can ask that question.
Okay, we can definitely help enrich, for specific sequences in plants and specifically in cereals. Cereals generally have a very large genome, and so there would be a lot of benefit in enriching, for specific or target sequences within cereal. But I think we would have to look at that on more of a case-to-case basis to see exactly what they wanted to enrich for and exactly what the goals were with their sequencing.
Right, it would kind of depend on the application, whether or not you're looking, if you're doing like genome sequencing, it's probably not necessary, but if you're looking at a specific region, they'd have to do a pretty custom design.
Yeah, we would definitely have to do a custom design for that, and as Ibrahim has mentioned, that's something we can definitely help customers with. We can help walk you through how to design these things, and we can give some advice on parameters that should be used for the different types of design in different organisms.
Okay. So Ibrahim, I'm going to give this question to you.
Sure.
You've kind of touched on this already, but can you supplement the SureSelect RNA baits with IDT synthesized xGen probes?
Yes, you can. In fact, the example that I showed earlier in the presentation was an RNA-based capture technology using RNA reagents. The only caveat I would say is that we've had some information in which when you're using RNA-based reagents, you just want to ensure that the stringency of your wash buffers are not too high for targeting anything below 30% GC. However, with RNA-based technologies, the problems generally occur with high GC content regions. And so if you're trying to supplement an RNA capture, you shouldn't have a problem with the reagents that are supplied with that manufacturer. It's only when you have below 30% GC in which you would need to reduce the stringency of your wash buffers.
Okay, good answer. Here's another question for you, Ibrahim, I guess. So does IDT help with the repetitive region workaround if somebody needs to design around repetitive regions?
We do not have the repeat masker database in-house as of yet. This is something that we're working on. Our website, the support page on our website actually has instructions on how to mask for repeats, and then the type of information we would need to tile around those repeat regions.
And I would just like to add, we can also assist customers directly, too, if they have questions regarding repeat masking.
And they would email, who would they email? Would they email technical support for that?
They would email xgen@idtdna.com.
Okay. All right. Adam, here's a question for you. This is getting a little bit away from the target capture, but it's also still enrichment. If you're doing PCR for enrichment, how many cycles can one do of PCR in order to minimize the bias arising from PCR enrichment?
There's, this goes along with a lot of the optimization that's needed for NextGen. There's not a great answer for that right now. And part of that depends on how much starting material a person has. So what you want to do is you want to, obviously you want to minimize the number of cycles that you're using for PCR, but you don't want to have so few cycles that you don't end up with enough product to actually sequence. So it's a matter of trial and error, and what I would encourage most people to do is to test the amount of material they have, and different manufacturers for different next-gen platforms have different ways of quantitating the amount of material that's needed. But by following the manufacturer's instructions, test the amount of material that you have after PCR. If that's not enough material for the sequencing run, then you obviously need to do more cycles. And if there's excess material, then you can cut back on the cycles. But it's really something that doesn't have a clear answer of a certain number of cycles.
You sort of want it to be the minimum in order to get the data that you need, basically.
Exactly. Yep.
Yeah. Okay. So the next question that I have for you, and Adam, I guess I'm going to go to you with this one as well, which is, are there any rules, guidelines as far as the size and type of DNA fragments that you can use the xGen probes with?
No, not really. The rules really regarding the size and length of the probes has more to do with the platform that you're using and the actual type of sequencing that you're doing with the platform. So, for instance, the 454 sequencers you sequence a fairly long amount of material, so you can have larger fragments with that and still cover the complete sequence. At the opposite end of the spectrum, solid has very short sequencing runs, and so you need to have very small fragments of DNA to run in that. But as far as the enrichment goes, it's not really it's not as sequence dependent as the other steps in the next-gen sequencing are. So follow whatever the manufacturer's instructions are for your machine is, I guess, the simple answer there.
Okay. And actually, I think I'm going to give this method question to Ibrahim. It's regarding enriching for GC-rich regions and basically performance of xGen lockdown probes. How do they perform for enriching for GC content?
I think our probe technology that's where that is actually where it excels at, is the high GC regions. So the coverage plot I showed you earlier in the presentation, that increase in performance is actually a GC-rich region. And the reason for that is that we QC the probes before we ship it out. So we verify the presence of a GC-rich targeting sequence before it's shipped out.
Okay, and actually I'm going to skip a couple questions here, and I'm going to actually ask you a question that's related to that, Ibrahim, then, is how are the xGen lockdown probes different from other target capture probes?
Sure, thank you for that. So other target capture technologies are derived from an array, in the sense that there's various techniques to synthesize probes on an array. I think some lithography techniques, and essentially those baits, as they're called, are synthesized in parallel in femtomolar quantities, and then they're cleaved off the array and shipped and put into a tube and shipped to the customer. So essentially, it's virtually impossible to QC the presence of any individual probe sequence. And so this is why, and naturally speaking, synthesizing certain sequences is difficult than others, particularly high GC sequences or extreme GC content sequences. They're just very difficult to synthesize. And so if there's no control for that, you're just taking it, you're just hoping that the presence of an individual sequence is actually present in the pool. So xGen lockdown probes, on the other hand, are individually synthesized and QC'd. We do not synthesize them on the array. And so we can verify the presence of each sequence in molar quantity, in molar purity, before we pull them or before we send them to the customer. And furthermore, that individual synthesis process yields a lot more material per probe. Rather than femtomolar quantities per oligo, you have a minimum of 2 picomoles per oligo. So that's magnitudes of more material per oligo that you receive.
Okay, great. Here's another question. I guess I'll give this to Ibrahim first, although Adam might have something to contribute to this. For exon capture stuff, is there a database that we recommend for looking at exome sequences and designing?
At the moment, we feel the easiest way to extract exon sequences using Ensemble. However, we feel more if you are okay with us extracting the sequences, we feel more comfortable with NCBI because of the version control that they have with that. However, we have some instructions if you want to go the Ensemble route and how to extract those sequences and send them over to us.
So again, that would be something where it would probably be best to contact someone directly.
Definitely, yeah.
Before.
Yeah.
So
that's right.
xgen at idtdna.com, you can email that. Okay, and kind of on a related note here is, and I don't know if there's a standard answer for this, but how much is the charge for custom design help?
Adam, you want to take that one?
Sure. Yeah, the custom design help that we offer comes with a variety. There's a variety of levels of help that we give different customers on a variety of different issues. And our general rate that we charge is, well, for the first 2 hours of design work that we do, we will do for free for customers, and that's per project. Beyond those first 2 hours, we generally charge $40 in the United States for each additional hour of design time. As I mentioned, there's a variety of levels of design that we have, and we can speak with individual customers regarding their specific applications as to how much time and how much money that would cost. And we can work a variety of different levels with customers too, depending on how much input they want to have on the design, how much work they want to do on their own, and how much work they want to pay us to do. So we're pretty flexible with that, is the short answer.
A little bit of a clarification on that, Adam. This is something I'm not sure about. Is that 2-hour design minimum, that first 2-hour design, does that only include if they purchase the actual panel? Or is that just for everybody, they get 2 hours?
Right. Generally, for everyone, we'll give them 2 hours. And especially in these types of situations where there's a lot of this is a relatively new technology, and we're relatively willing to, help people out on this without having them purchase something up front to make sure that it's going to work for them and to make sure that it's going to be the best option that they have. Now, for larger projects that are going to take more of a time commitment for us, we do like to work out some type of purchasing agreement ahead of time. But again, we can discuss that individually with anyone who has any questions or concerns about it.
Okay, great. Here's a pretty straightforward question. Do we have any software? Does IDT have any software for designing the panels? Ibrahim? Adam? Here, I guess.
The short answer, sure. The short answer is no. We don't at this point in time. It's something we're working on internally, but our feeling is this technology and next-gen sequencing in general is such a developing field at this point that trying to develop a technology, trying to develop software that's going to work well with this is just something that we're not comfortable releasing at this point. We do have a very large and very dedicated team of in-house bioinformaticians that can assist in any custom designs that are needed.
Okay. Ibrahim, did you want to add anything else to that?
No, that's fine. And I was just going to add that this is something, like as Adam was mentioning, the technology is so new that IDT would just like to validate any design algorithms before we do release that. And so that takes time. And it is something that we are working on, though.
Okay. Ibrahim, I believe this is a piece of data that you have. It's something you've commented on before. In combining different panels, so if you're supplementing something with the xGen lockdown probes, what concentration of the probes do we recommend?
There's various answers for that. We have one customer that would start with 13 picomoles and take a fraction of their supplement size versus their overall capture size, and that is the amount of material that they use to spike in. So, for example, if you have a 200 kilobase supplement size, and then your overall capture panel is 1 megabase, you would need 0.2 times 13 picomoles, which is, sorry, my math is terrible in my head, but
we probably don't need the exact number.
well, I guess that's the point. It is a fraction of the supplement size over the overall capture size. We have other customers who just do a 1 to 100 of lockdown probes to whatever capture panel that they're using, whatever capture technology that they're using. I think the gist of it is that you want a smaller amount of lockdown probes than you do of your overall capture. And the more you add, the more enrichment that you get in those regions of interest that are troublesome.
Okay, so it's the final concentration isn't critical then?
No.
Okay. All right. So the next question here is well, I'll ask this question, so it's up to Adam. Have we done any sequence capture underlying QTL in plants? And what are necessary for the successful QTL sequence capture? QTL, is that familiar?
I believe that they're referring to quantitative trade locus.
Oh, right. There you go. Thank you.
And no, we haven't really done a lot of work with any of our collaborators who work with plants. Most of the people that we've worked with are working in mammalian model systems, and we haven't really moved into plant systems yet with this. So unfortunately, I don't have a lot of data that I can share with anyone on that.
Plant stuff's got to be coming with all the agriculture stuff.
It is, yeah. And we definitely have some people that are interested in it, but unfortunately, we just haven't had the chance to collaborate with anyone on anyone in the plant field at this point.
All right. So here's a pretty specific question that I'm going to give to Ibrahim. There's actually two of them that are kind of related. So one of them is whether or not the cost of xGen lockdown probes is comparable to SureSelect. And then this other person has ordered two 500 KB designs that are SureSelect, and they're getting ready to order a third one. And they're wondering if there'd be a price difference as well as how would the delivery time be compared to SureSelect?
Sure. So the pricing of lockdown probes is very different from how you would price a SureSelect product. We charge per probe. And so for, I will give you USD list pricing. But for our mini scale, it's $15 USD. For the standard, it's $25, and the XL is $35. And then because we charge per probe, it just depends on how many probes you need, and that's dependent on the size of your capture. And so, if your panel is 1 megabase, it will probably be much larger amount of upfront cost than a SureSelect panel. But for smaller capture sizes, you will have a decreased amount of investment. Overall, though, if you use up all the material, the cost per enrichment would be about $100 for the mini scale product, $15 for the standard scale, and less than around $1 per enrichment for the excuse me, I have something in my throat, for the large scale product, the XL. And that's if you use up all the material. And so the real cost benefit comes with as a per sample basis. And as far as the delivery time, the product will be shipped within 7 to 10 business days of your order. And I think there's one more question regarding the 500 KB. And I guess, so he has purchased, he or she has purchased two 500 KB panels. And they have one 500 KB panel. And I think it just depends on, is this panel being used to supplement their existing one? And I would encourage them to review what the designs that they have in place and why they're purchasing it. And I think that will ultimately determine whether or not they should go with the lockdown probe product versus a rate capture product.
It kind of doesn't look, just from reading the question, it doesn't look like they are supplementing with this. It's just a 500 kb design, and
Sure.
this will be their third order of a 500 kb design. I don't know if they're reordering the same thing or if that's a brand new one each time.
Sure, sure. The final, I guess the only other thing I'd add to that is with depending on the sample volume. If you have hundreds or thousands of samples, you could definitely benefit going with the lockdown probe approach.
Okay. I'm going to throw this out to Ibrahim first, but I think, Adam, you could probably add something to this as well. So how many targets can you do per reaction, and then how do you process the samples? My guess is they want to know just the target capture portion of the sample. So, like, how do you actually do the capture?
So I'll take a shot at that one. In terms of capture size, I do not think there's really an upper limit. I know the largest capture panels you can purchase today go up to 60 megabases, even higher. So it's not about, to me, it doesn't seem like there is an upper limit to the capture size that you can have. And as far as the protocol, our product is been shown to be compatible with commercially available kits today. We don't offer an assay right now or a kit to go along with our product as of yet. However, commercially available reagents such as the CCAP reagents or the SureSelect reagents are compatible with our product. And the only caveat being that with the SureSelect reagents, I would encourage reducing the stringency of the wash buffers if you're targeting regions below 30% TC.
Okay, and let's see here. There's one more question here, and this question is, I guess I'll direct this to Adam. So NCBI is not comprehensive for variants. Is Ensemble better than NCBI or the UCSC genome browser? And then also, do we have any information about gene code?
About gene code?
Kind of a bunch of, apparently that's another database.
I'm sorry, that's a database I'm not familiar with.
Okay.
But yeah, as far as looking into variants, NCBI and Ensemble are both pretty much about as good as we have right now. Particularly when you get outside of humans, there's not a lot of annotation and it's fairly sparse. The genome browser is a little bit behind, and the big problem with genome browser is there's not a good method of they don't annotate well how often they're updating the databases that they're using. So for instance, in comparison, if you're using the NCBI databases, and particularly if you're using the RefSeq databases, there's a very nice versioning system that's put into place that allows you to see, what version you're on and make sure that you're comparing the current version to the current version. That doesn't exist in the genome browser, to my knowledge. So I think using NCBI and Ensemble are the two most up-to-date databases that there are currently.
Here's kind of a database-related one, too. When you're designing your xGen lockdown probes to supplement a exome capture or whatever, do you have to run a sequencing experiment first to see where to design your exome, your xGen lockdown probe set, or is there like a database that you can go to and see like where the coverage is good and where it's not?
Well, it's sort of a trick. Sorry, I guess Adam, you can correct me if I'm wrong, but I think there is databases available, particularly the 1000 Genome Project, which provides annotated information on where sequences have been covered and not been covered. However, the different capture sets that come out have different versions as well, and so I strongly suggest doing a sequencing run first before trying to supplement for it, but the pattern has traditionally been first ones, high GC content regions are not captured very well.
Yeah, and I would agree with that as well. I think there's not a great way to determine what you need to supplement until you can see what deficiencies you have. So and part of that can vary based on the actual conditions that you're using for the hybridization and for the enrichment. And part of it can also just simply vary based on the cell line you're using, the DNA preps that you're getting from that cell line, and things like that. So there's a lot of variables that go into it, and I would agree with Ibrahim, and I would strongly encourage anyone to do a sequencing run first, see where their problem areas are, and then design enrichments from that.
Okay, and at this moment, it does not look like we have any additional questions. If anybody wants to type in one more really quick, but otherwise I think we're done. I guess we're done. So, all right, at this point I wanted to thank Ibrahim and Adam for their time and giving this presentation and answering questions. And I want to thank everybody for attending the presentation today. So this is one of a series of webinars that IDT will be doing on next-generation sequencing target capture, as well as a bunch of other topics on qPCR and synthetic biology. And we've recorded today's presentation, and we'll make that available on our website at www.idtdna.com. And you'll find that under our support tab under our technical and educational materials. And then it'll also be posted on our YouTube channel, which is www.youtube.com/IDTDNABio. You'll find this webinar there, as well as some previously recorded webinars on those other subjects, qPCR, next generation sequencing, and synthetic biology. All right, and at this point, I guess we will call it good, and I will say best of success in your research, and thanks again.
Thank you.
Thanks, everyone.